This is why septic shock causes lactic acidosis. This is why hypoxia and ischemia shuts down oxidative phosphorylation in the Krebs cycle. You can clamp an artery, you can cut out a tumor, but if you don't understand how the cell makes energy and what can stop it, you're going to miss what's killing your patient. Today, we're going to go through cell biology for surgeons. And if you're a surgical resident studying for the ABSITE or your board exams, this video is going to get right to the heart of all of those details. So, cell biology for surgeons today at CitizenSurgeon. Let's go. You're in the ICU. Your patient is in shock. Lactic acid is rising. Why? You're hypoxic. There's no more oxygen. Mitochondria is shut down. Glycolysis is running on fumes. In today's video, we're going to go through cell biology. We're going to talk about the cell itself. We're going to go through all the organelles. All right. We're also going to talk about DNA replication, transcription, translation, why that's important for surgeons to know. And then we'll go through metabolism at the end. Along the way, I'm going to have some multiple choice questions. Check that one out. All right, I'm going to have some clinical vignettes to keep you interested in and make sure you understand that this is absolutely clinically applicable. For those of you studying for the ABSITE or your board exams, I'm going to be taking you on a journey. If you haven't bought the book ABSITE Review by Fiser, I highly suggest you buy that book right now. I got a link to it in the description below. And I'm going to follow the table of contents of that book until I get to the end. All right, so today is cell biology. That's chapter 1. You can follow along with me. I'm covering what Fiser covers, but I'm also including those things that he left out. And I may be skipping over the things I didn't think were critically important. So, cell biology for surgeons today. Let's get right into it. And where do we start? We start with the cell membrane. So, what is the cell membrane and why is it important? Well, the cell membrane, as we know, it's a phospholipid bilayer. It contains cholesterol, protein channels, as well as enzymes. Okay, it maintains a ion gradient across so that we have a negative inside of the cell. So why is all this important? Well, check this out. There are several problems that we see in surgery. For example, pemphigus vulgaris or bullous pemphigoid. I'm going to talk about what each of those are. Think about burns. When we have a severe burn, we're going to have disruption to our cell membrane and we get fluid creep and we get a loss of a significant amount of electrolyte. Cell membranes are important in cancer, and we're going to talk about that a little bit later. In organ transplantation, we have features of the cell membrane that have a significance in HLA compatibility. With aging, we get changes in our cell membrane, and of course, with blood transfusion, our cell membranes contain our ABO blood type. So in surgery, we're dealing with the cell membrane all of the time, and it's important to know what's in it. So take a look at this table, and here I'm going to show you some of the important aspects of the cell membrane. So number 1 is that it's a phospholipid bilayer. What are the testable facts? So it's amphipathic, also forms a barrier. And when we get damage to that cell membrane, so for example in burns, we can get fluid loss and electrolyte loss. Cholesterol is important in the cell membrane, and that modulates fluidity within the cell membrane, and it affects membrane repair and signaling. There are several transmembrane proteins. These span the bilayer and can include receptors and transporters, and these are crucial for the immune response, drug delivery, and of course cell adhesion. And we're going to talk about that in just a minute. Finally, we have peripheral proteins. These can be part of the cytoskeleton, signal transduction. These are loosely attached to the cell surface. And of course, glycoproteins and glycolipids. So these are important in ABO compatibility with glycolipids, and of course, HLA antigens on glycoproteins, which is important in organ transplantation. So those are kind of some of the initial important aspects of the cell membrane that you got to understand. Well, what about this transmembrane gradient? What is the thing that's responsible for that? Well, check out this diagram right here, and we're going to quickly go over the electrolytes and why it's important to know roughly which ones are higher on the inside and which ones are higher on the outside of a cell. So check this out. So the most basic thing to understand is that sodium is high outside the cell and is very low inside the cell. The opposite of that is potassium, which is very low outside the cell and much higher inside the cell. We also have calcium, which is much higher outside the cell than inside the cell, and magnesium, which is about equal but slightly lower outside the cell. Well, those are the cations. Let's look at the anions. So the most popular anion is going to be the big friend in salt, okay? So NaCl is chloride, and chloride is low inside the cell compared to very high outside the cell. We have bicarbonate, which is higher outside the cell, and then sulfate and phosphate as well. Now, what is the major actor when it comes to maintaining this gradient? Well, the major actor is the sodium-potassium pump, and that is going to pump sodium outside the cell and then bring potassium inside of the cell, okay? So why is it important for a surgeon to understand that? So we're going to get 3 sodium ions pumped out every time. We also have 2 potassium ions brought in, and that's going to use 1 ATP, and we're going to get to ATP in a little bit. But why is this important? So if we didn't have this pump, then sodium would follow its gradient. You'd have a lot more sodium inside the cell, water would follow, and all the cells would just burst. And that's osmosis, okay? So it's important to understand that. It's also important when we look at cardiac conduction and nerve cells because the resting membrane potential depends on this ion gradient. Well, what else? Well, we also have transport of ions and molecules across a barrier. So we might have a hydrogen-potassium transporter. We might have a glucose transporter that's dependent on a sodium gradient. Also, there's a small amount of heat production. So sodium-potassium pump produces a small amount of heat, and during shock and hypoxia, where we get decreased ATP and decreased function of this pump, we have a hypothermia risk. Then finally, after a reperfusion injury, after ischemia, the sodium-potassium pump helps restore ion balance. So for a lot of things that we may take for granted every day in the ICU, this transmembrane gradient provided by the sodium-potassium pump is critically important. And so let me quickly take you through the different ions and what their surgical relevance would be. Check this out real quick. So we have sodium. So sodium, the main location is extracellular. It drives fluid shifts. It's important in resuscitation and, of course, in syndrome of inappropriate ADH after neurotrauma. We have potassium. That is primarily intracellular. This is critical for cardiac rhythm. Hypo and hyperkalemia can cause fatal arrhythmias intraoperatively and postoperatively. Calcium, it's mostly extracellular, so it's very, very small concentration intracellular. It's needed for coagulation. It's needed for muscle contraction. It's needed for signaling. We have low calcium levels, we can get tetany. We can also get bleeding. When we look at magnesium, this is primarily intracellular, and this is critically important when it comes to the function of enzymes, also stabilizing that transmembrane gradient. Low magnesium levels can worsen the arrhythmias associated with hypokalemia, and also low magnesium can make hypokalemia difficult to correct. And when we look at chloride, bringing it back to the table here, this is primarily a extracellular ion, and chloride follows sodium. It has an acid-base role. We can get a chloride shift, especially after massive transfusion. When we look at protons and bicarbonate, this is central to acid-base balance. Metabolic acidosis and alkalosis certainly affects our clinical outcomes, and phosphate is primarily intracellular. It's needed for ATP 2,3-bisphosphogluconate, or 2,3-BPG, is an important aspect of the glycolytic cycle, and that is needed for ATP production. We find that phosphate can get very low in refeeding syndrome as it is sucked up by the production of ATP. So those are the ions. You should probably freeze frame that slide and keep that in your study Rolodex so you have it for fast reference. All right, so let's get to a clinical vignette. What do you think about this question? So we have a 32-year-old man who was brought to the trauma bay after a high-speed motor vehicle collision. He is in shock. He's requiring massive transfusion of packed red blood cells and normal saline. After several hours of resuscitation, we get another repeat set of labs, and it shows the sodium 142, potassium 4.3, chloride of 115, bicarbonate 18, pH is 7.28, and his lactate is 6.8. Which of the following is the most likely cause of this patient's metabolic acidosis? So is it lactic acidosis from tissue hypoxia? Is it hyperchloremic acidosis from excessive saline infusion? Is it renal failure due to ischemia? Or is it intra-abdominal hemorrhage with shock-induced acidosis? So when we look at these answer choices and we think about the ion discussion that we just had, which of the answer choices is most likely? Well, I'll pause for about 2 seconds. Well, you could push pause if you wanted to. And the answer is hyperchloremic acidosis from excessive saline. So when we look at normal saline, it has 154 milliequivalents of sodium, it has 154 milliequivalents of chloride, and our normal chloride is between 104 and 111. That's going to drive our chloride up, and that's going to help create a metabolic acidosis. Okay, so number one, it's critically important to know what the ion levels are in your bags of resuscitative fluid. Okay, so for example, what is the electrolyte content of lactated ringers? And that's an important question, especially if we're going to resuscitate a patient that has a head injury. We might make them hyponatremic because lactated ringers has a lower concentration of sodium ions. So ions are critically important, whether we're in the ICU or the trauma bay or even daily rounds on our patients. Let's get back to that cell membrane. And I gotta say something. I gotta ask, if you like this, give it a like, give it a share. Sign up for notifications. Check more videos out at CitizenSurgeon. I love having you here, and leave a comment. I love it when you're asking questions. It drives me to create more value, more videos, and when you ask an interesting question, it makes me go to the books and come up with a good solid answer for you. So, all right, let's get back to the rest of the cell membrane. So we talked about the ion channels, and now it's desmosomes, hemidesmosomes, and of course, proteins and lipid rafts. So what about desmosomes and hemidesmosomes? Well, the important thing to think about is, well, which ones, what do they do and which ones which? Well, they help in cell adhesion. So I like to think that desmosomes, that is cell to cell, whereas hemidesmosomes, that is cell to basement membrane. So if we have weaker desmosomes, we get decreased cell-to-cell adhesion. If we have weaker hemidesmosomes, one of which we see in aging, we get inability to adhese that cell to basement membrane, and we see, as in aging, thinner skin and the ability to shear the skin much more easily. So those are effects of weakened hemidesmosomes. All right, I think we may have exhausted the cell membrane when it comes to surgery. I think we've covered most all of that. And so let's get to the next part of cellular biology for surgeons, and that is the cell cycle. So we know that a cell is in a cell cycle. We're either in G0 phase, which is just kind of the quiescent phase, hanging out, or we're in this active cell cycle, and it's broken up just like this. So check this out. We're going to start with G1. So what is G1? So G1 is our gap 1, and this is important for cellular growth. This is where we're creating proteins, we're creating energy so that we're ready for DNA replication. And this is all occurring in G1. If cells are damaged after trauma or after surgery, it's likely that they're going to stay in G1 for repair until they're ready for DNA replication. Now, what stops it there? I'm going to ask you in a minute. So, what is the cell cycle checkpoint? And we have a checkpoint each time we're going to a different phase of the cell cycle. So, what arrests a cell from going to the synthesis phase? What will keep that cell in G1 if we have trauma or damage or something? What is the, let's say, tumor suppressor gene that acts to arrest the cell in G1? We're going to talk about that in just a minute. So let's get to S phase. Well, what's happening at S phase? S phase is synthesis. This is where we have DNA replication. So when we get DNA replication, this is the duplication of our chromosomes. And why is this clinically important? Well, many chemotherapeutics act on cells during this phase by targeting DNA replication. Good examples of that might be 5-fluorouracil or methotrexate. These are going to target DNA replication and stop dividing cells, for example, cancer cells, from dividing. After we get done with synthesis, now we go into G2. And if we're in G2, what is the cell doing? Well, the cell is going through a series of checks to make sure it's ready to split. We call that mitosis. That's where we get a splitting of the cells after DNA replication. If we have injury to the cell or we have some genetic damage, then we will stay in G2. So the hope of 5-fluorouracil or methotrexate is to cause damage to DNA in a cell so that when we get to G2, the cell arrests and it doesn't divide because there is damage within that cell. So we get cell cycle arrest, and that is the goal of radiation. It's also the goal of chemotherapeutics. Now, the next phase is mitosis, and this is where we get the splitting of the cell. Now, again, between G2 and mitosis, we're going to have another cell cycle checkpoint. I want you to think, what is the cell cycle checkpoint when it comes to getting from G2 to mitosis? Now, one thing we should go back to is G0, so this gap zero phase. So can you tell me which cells are normally in G0? They spend almost all of their life in G0. Well, this would be neurons, skeletal muscle, also cardiac muscle, and this is important because these cell types are very resistant to healing, okay, because they're not in an active cell cycle and getting active turnover. And we think about this in nerve injury. So if we get a significant injury to a neuron, it's going to take a significant amount of time for any healing or regeneration. So let's get back to these checkpoints. So G1 to S, what are the checkpoints there? So that's P53. So we call P53, it's a tumor suppressor gene, right? So it suppresses tumors because it's looking for damage and it's going to cause cell cycle arrest. If we get a mutation in P53, it can no longer look for damage and it's going to just push cells straight from gap 1 to synthesis, and then we're going to have replicating cells, and that's what we see in cancer. So P53 and retinoblastoma protein are two tumor suppressor genes or two cell cycle checkpoints from G1 to synthesis. And that is very important to understand, especially if we're learning about chemotherapeutics in cancer. What if we asked ourselves, is there anything that can promote a cell to go from G1 to S? Well, yeah, we have growth factors. So vascular endothelial growth factor. We see this after injury with capillary growth. Also platelet-derived growth factor. That can push cells from G1 to S phase. So when we look at things like the metabolic response to injury, I talked about that in the very first video I did. You can check that one out right here, where we talk about, well, what's the body's response to injury? Well, one of the things is we get profound circulation of cytokines and inflammatory markers and growth factors. And those are response to injury, and that can lead to cell repair, can lead to cell healing, and of course, increased cell turnover. Well, let's look at the other important checkpoint, and that is from G2 to mitosis or cell splitting. What are the important cell cycle checkpoints there. Well, these are the cyclin-dependent kinase, or CDK. And there are two clinically important things with cyclin-dependent kinase. Number one is radiation or radiotherapy in cancer typically arrests cells in that G2 phase. And second, it's important for wound healing. So we can get delays in wound healing because of cyclin-dependent kinases resting cells in that G2 phase. So let's go to another clinical vignette. Let's say that we have a 65-year-old male who presents with painless hematuria. He has a CT urogram that reveals a 3.5 centimeter mass in the renal pelvis. Biopsy shows transitional cell carcinoma. Immunohistochemistry is positive for a p53 mutation, and the patient is started on cisplatin-based chemotherapy. The question is, which of the following best explains the mechanism of action of the chemotherapy used? So, inhibition of DNA unwinding and transcription, cross-linking of DNA to prevent replication and transcription, interference with mitotic spindle formation, or inhibition of ribosomal translation. These are the type of questions that are going to come up on the ABSITE, on board exams, for instance, for medical students, perhaps even on the shelf exams. So if we can really understand the cell cycle and link chemotherapeutics to each of these phases or each of these arrest points between cycles, it's much easier to remember them. We don't have to remember a whole list anymore. So the answer to this is cisplatin cross-links DNA to impair both replication as well as transcription. That usually leads to cell apoptosis, or programmed cell death. And this is an example of why it's really important to understand these processes. And so, talking about transcription and translation, why do surgeons need to understand the nucleus, transcription, and translation? Well, we need to understand this for things that I've already talked about. So cancer biology, targeted therapy, understanding cellular stress, understanding the mechanism of action, not only of chemotherapeutics, but things like antibiotics, and also understanding inherited syndromes. So mitochondrial syndromes or storage diseases, lysosomal storage diseases. If we can understand the different aspects of the cell and cell biology, it's much easier to remember and understand what's happening in our patient with a particular inherited syndrome. So let me outline this very simply. We got a nice pink cell here. In that cell, we're going to have the nucleus, okay? So that has a double membrane, and this is where DNA replication as well as DNA transcription, or going from a double helix DNA to single-stranded RNA, happens. When we think about the cell cycle and we think about replication and transcription, there are a number of different growth factors which are going to be important in these processes. Now, I'm not going to go over all of them, but you can see them right here. So p53 is very important, going from G1 or GAP1 to synthesis. When I did the colorectal cancer series, I talked a lot about that adenoma to carcinoma pathway, and p53 was a big part of that. We have the MYC proto-oncogene. We have NF-kappa B. We have HIF-1 alpha, hypoxia-inducible factor 1 alpha. With respect to breast and prostate cancer, we have estrogen receptors, we have androgen receptors. FOXO is related to oxidative stress and metabolism, and OCT4, SOX1, and NANOG. This might ring a bell as Yamanaka factors when it comes to creating pluripotent stem cells. So this is just a list of growth factors came up on your exams, also important for targeted therapy and different actions within metabolism and cellular division. Okay, let's look at the rest of the cell. So outside of the nucleus, the next most important thing is the rough endoplasmic reticulum. Now, single-stranded RNA is going to go across that double membrane of the nucleus. It's going to go into the rough endoplasmic reticulum, and this is where we're going to get the formation of secretory proteins, okay? Proteins that are going to be excreted. But we have a lot of other important organelles. We have mitochondria. This is the site of oxidative phosphorylation. This is where we're going to take oxygen and we're going to take the products of the glycolytic cycle and we're going to make a ton of adenosine triphosphate, or ATP. What are some important things about mitochondria? Well, we get maternal mitochondria inheritance. Those are things that are passed down from mom to the fetus, and that mitochondria is preserved. We also, when we look at cyanide poisoning is basically shutting down mitochondria, and also linezolid, the gram-positive antibiotic, works to shut down mitochondria in those gram-positive bacteria. So those are some good clinical tie-ins when it comes to this important organelle, the mitochondria. We talked about the rough endoplasmic reticulum. That's the little oval there with all the purple dots. So secreted and membrane brown proteins are going to be created by the rough endoplasmic reticulum. We call that translation. That's when we're taking single-stranded RNA to proteins. Where do we get a ton? What cells do we get a ton of rough endoplasmic reticulum? Think about secretory cells like plasma cells, okay? B cells for antibody production. They're going to have a ton of rough endoplasmic reticulum. When we look at smooth endoplasmic reticulum, what cells have a lot of that, and what's involved? What is smooth endoplasmic reticulum important in? It's important in lipid synthesis. It's important in detoxification. And it's also important in calcium storage. We see liver cells or hepatocytes have a large amount of smooth endoplasmic reticulum. So when we look at these yellow G organelles, that's the Golgi apparatus. Why is that clinically important? Well, these are important in post-translational modification. So if we have defects in our Golgi apparatus, we can get impairment in protein trafficking disorders. We can get glycosylation defects in several other congenital problems, and these have to do with an impaired Golgi apparatus. When we look at the lysosome is kind of digestion within the cell. We see lysosomes, or the phagolysosome, is important in digesting bacterial cell membranes and bacteria contents. There are a number of lysosomal storage diseases like Gaucher's disease and Tay-Sachs disease. So this organelle is very important in those patients. The P cell is the peroxisome, and this is important in long-chain fatty acid oxidation. It's also important in detoxification. And then finally, those little lines, that is the cytoskeleton, and of course that is really important for cell structure. Now, to finish cell biology for surgeons off, let's get to one more clinical vignette. Here we have a 55-year-old man who's admitted to the ICU after a bowel resection for ischemic colitis. He's tachypnic, he's hypotensive, his labs show a lactate of 6.2 millimoles per liter, a pH of 7.23, a bicarb of 18. What metabolic pathway is primarily responsible for his elevated lactate? Well, to understand this question, we got to talk about the last thing today, and that is metabolism. Oh my God, metabolism. This medical school has took like 2 weeks to go through. Well, we're going to do it in about 1 minute, okay? So metabolism is how we get glucose to energy, okay? Glucose to ATP. Well, how do we do that? So to get through it quick, check out this diagram right here. So we have glycolysis. That's going to take glucose to pyruvate. That's going to make 2 ATP. We're also going to get 2 NADH from that. And now we're going to get to the mitochondria. So what's happening in the mitochondria? Well, the pyruvate that's broken down in glycolysis goes to the mitochondria. We get pyruvate oxidation to acetyl-CoA. That enters the Krebs cycle, and we get a couple of more ATP with some CO2, as well as 6 NADH and FADH2. NADH and FADH2 are going to go through that transmembrane within the mitochondria, where we will have massive AT production. We're going to get 30 to 34 ATP to be produced from the byproducts that are coming from the breakdown of one glucose molecule. And what do we need to do this? We need oxygen. You can see this coming in from up top. We have glycolysis, the breakdown of glucose. Krebs cycle is the oxidation of acetyl-CoA. And of course, gluconeogenesis is where we take pyruvate or things like alanine or glycerol from the breakdown of fatty acids, and we make glucose again, so that's gluconeogenesis. And it's important to understand that we have those metabolic pathways. Well, Are they upregulated, downregulated, or just static in different states? So how about the fed state? Or how about the starving state? Or how about after alcohol ingestion? What do these look like? Well, check out this table. I think you should screenshot this one as well. So in the fed state, our glycolysis is up. Insulin is going to promote this. Our Krebs cycle is up. We're going to be polishing off that acetyl-CoA, and gluconeogenesis is downregulated. In the fasting or in the stressed state, we want to preserve all of these building blocks. We want to shut down glycolysis. We want to shut down the Krebs cycle, and we're going to promote gluconeogenesis to the formation of glucagon and cortisol. What about in the hypoxic state or the shock state? Well, we're going to do glycolysis, but now glycolysis is anaerobic. Instead of going to pyruvate, we're going to be forming a lot of lactate, okay? And that lactate is going to build up when we do very heavy exercise and we get rid of all of the oxygen in our muscles. We start building up lactate and that burns. How do we get lactate back to pyruvate? Well, we go up to the liver where we go through the Cori cycle, okay? And that's important when it comes to lactate metabolism. But back to this table. So in the Krebs cycle, because we have no oxygen, we've shut that down. We're not going to be metabolizing any acetyl-CoA. And gluconeogenesis is also shut down because we're in shock and we don't have any oxygen. How about after alcohol intoxication? Well, we have increased glycolysis, so we have increased NADH. We are anaerobic in our cells, so we have increased lactate production. Our Krebs cycle is shut down and gluconeogenesis is shut down. So these are some of the changes that happen when we have different states with respect to each of those glycolytic pathways. So let's get back to that question. What metabolic pathway is likely impaired in this patient with the elevated lactate? Well, the answer is anaerobic glycolysis. This is the primary reason for the elevated lactate because in shock, tissues are hypoxic, forcing glycolysis to proceed without oxygen, producing lactate as a byproduct via lactate dehydrogenase. Okay, so today, a heavy day, all right? Cell biology for surgeons. There's a lot to know. There's a lot to remember. If you're studying for the ABSITE, your board exams, your shelf exams, I think we definitely covered the highlights, and I tried to tie them in to clinical vignettes and clinical syndromes to help you link this so you're just not trying to memorize a list. If you understand the why, you will remember it always. You don't have to remember lists. So, what did you find most helpful? What did you find most confusing? Leave a comment below. Definitely, I'll enjoy engaging with you and getting back to you. Other videos to check out the metabolic response to injury, and also check out my nutrition videos. Those are going to kind of bring these cell biology topics and cell biology learning points really to the forefront and help you remember them. So check those videos out. As always, stay safe, study hard. I'll see you next time.